Abracadabra. I want to reach out and grab you. Abracadabra. I want to reach out and grab you. Hey, what's up, weirdo? What's going on? Nothing. I can't get this, get my do now up. What do I gotta do to get my do now up here? I gotta.
Hi, Mr. Pope.
Hey, what's up? I'm
Hey,
trying.
Mr. Pope.
Hey, what?
Last class, how comes it said I was removed from meeting by host?
Because I removed you because you didn't respond.
I know. I sent you a message. I was messaging you.
Yeah, I saw it later. So
Uh,
you want to go TT right now in case I call on you again?
no, I was there. I could hear you. You moved me. You came to my break. Not breakout.
Oh, I
You
never
moved to the private room.
respond. I never got you to respond to say squat whenever I was talking.
I know, because my mic wasn't working.
Oh, well then you never chatted. Did me ask you to chat too? I said, can you chat and say something? No.
You didn't get my message?
Nope.
I was sending you like 3 messages. I messaged your phone number as well.
I did. No, later on I did see that, but that was after the fact.
That's fine then.
I'm trying to. Oh, there we go. Now maybe this is going to work. Let's see if this will work now. I'm fighting a losing battle here. I'm fighting a losing battle. I'm tired of fighting losing battles too. I don't have time for this stuff. Maybe what do I need to do? Why can I not get? I guess class hasn't started yet, so I'm not too tripped out yet, but we'll see. Why can I not see this? I need to open it. I've got a PowerPoint that I'm trying to display and I can't. So we'll see if I can eventually get it. Okay, now let's try this. There it is. Now I'm displaying it. See if I can get there. There we go. All right, everybody should be able to see it. Now I'm going to see if I can project it for the entire class here as well. See what this does. Is that going to show up there? Does not show there. Huh. Oh, well. I guess I'm going to have to just do it the old-fashioned way. All right, you guys in class? You in class, kids? Not yet. Woo! Everybody's getting too close to me, man.
You got close to me.
No, it's a lie.
I was
That's a lie. There's Javier Garcia. Do not put your answers into the chat yet. Do not put your answers into the chat yet. You might want to maximize that screen so you can see better. I don't know if you guys can see that. You did? You see that list? Oh, here it is. Anybody have a joke to make me laugh?
Sorry, rush out.
Do what?
I'm fresh
Let's
out.
hear it. What's your joke?
I'm fresh out of jokes.
You're fresh out of jokes? Come on, lies, man.
All I got are dad jokes, and no one wants to hear those.
What?
All I have are dad jokes. No one wants to hear those.
Oh, we all want to hear it. Give me a dad joke as long as it's not vile. I'm down with dad jokes as long as they're not vile. No vile jokes. There's got to be something. There's got to be something. Hey, what's up? Do what? That thing's not gonna work. I'm gonna have to do the user screen. Okay. This is for you. Grab one. This is for you. Grab one. Woo-hoo. This is for you. Grab one. Woo-hoo. This is for you. Grab one. You grab two.
But somebody in the chat said, Jalen said, what has a bed but doesn't sleep?
What has a bed but doesn't sleep? What has a bed but does not sleep? I don't know. Let me think about this for a second, though. Let's just not give up on it immediately. What has a bed? Vampire? Jalen, is it a vampire?
Now,
What is it?
wait, you actually don't know?
I actually don't know. What has a bed but doesn't sleep? What?
A riverbed.
Oh my God. I was gonna say my bedroom. My bedroom has a bed, but it doesn't sleep either. But it doesn't. It's not awake. But neither is a riverbed. Never was
Oh, I
awake
have a
either.
joke.
Well, let's hear it.
Okay. I hope it's not vile.
You hope it's not what?
I hope it's not vile.
Oh my gosh, I don't know if I can take vile. Okay.
What is the fastest way to determine the sex of a chromosome?
You're not gonna. This is not gonna be sexist in any kind of way, right?
No.
Okay. What is the fastest way to determine the sex of a chromosome? Well, autosomes have no sex related to them.
Ask why.
Oh my God, are you kidding? Asking why? Yeah, that makes sense. Asking why. So then they
No.
could find out. Is that it?
No, you pull down its genes.
You pulled down. his genes, but-
my
yeah, but did you hear? But Jalen just said, ask why. Because that makes more sense to me because if it's a Y chromosome, then it's going to be what?
Oh,
I
that was
have-
funny too.
I
I'm
had
sorry?
I had the biology reason. She had the, the
Yeah, the biology reason's a really good reason. I like that one. That makes a lot of sense, dude. I mean, if you ask why, it's going to be a dude. That's perfect. I like that. I'm going to keep it.
Yeah, keep it. Put it in a lesson plan.
Yeah, I'm going to put it in a lesson plan. That makes a lot of sense. Huh? All right. We've got about 1 minute. In 1 minute, I'm going to stop, and I do not want you to put your answers into the chat yet. Wait and put them in privately to the do now that I have up. And let's say people start putting them in at 12:07. I want to get everybody in here first. Get everybody in here first. We'll start putting them in at 12:07. Now you guys can talk about them. If what you guys think are the answers. I'm definitely going to come from you four first. Okay, it is 12:05. Good morning. It is, what is the day? What is today's date? The 8th. It is the 8th of February, the day after the Super Bowl. And we have a do now.
Wasn't really much of a Super Bowl, actually.
It wasn't as much of a shootout as I thought it was going to be. I predicted 60 points cumulative, and we didn't even get close. We got close to 40, and I was rather surprised. I thought it was going to be a much higher scoring game. Tampa's defense was beast frigging mode, and Kansas, I mean, Kansas couldn't get a touchdown to save their life. And that was it. I mean, what are you going to do? So yeah, it wasn't what I expected. Okay, now, nevertheless, we have a do now. I do want you guys, everyone, to answer it. There are 5 blanks here. Put them 1 through 5, 1 through 5, 1 through 5, and answer them. Do not put them openly. Do not submit it openly. When you hear the alarm, submit your answer. I'm going to shut up.
Wait, in the chat? Is that what you're
Yeah,
saying?
in the chat, 1 through
Oh.
5. When you hear the alarm, submit your answer. Okay, now you guys in here, talk them out. Talk them out. What do y'all think 1 through 5 is? Blanks. And why? I'm gonna definitely be coming to you guys for sure. So you guys can talk them out. I wouldn't worry about that yet. This is just old stuff. Mitosis and meiosis. These kids. Y'all only have 1 minute. Y'all gotta talk your way through these. What do y'all think the first, second, third, fourth, fifth one is? I'm gonna mute it in here so they can be talking without giving you guys clues. You're gonna number 1, 2, 3, 4, 5, and what the answer is for this do now in 52 seconds. Don't put it in the chat yet. And keep it private. Okay, now you can drop your answers, drop your answers, drop your answers. Drop your answers. Let's see what you guys have. I've got 1, 2, God bless. This can be hard to keep up with. I have 2 that are correct. 2 are correct. 3 are correct. 4 are correct. 5 are correct. I still only have 5 people that are 100% correct. Now 6 are correct. I'm looking for 100% correct. Let's see if we can get to at least 8. No, 9. Last class got 9 correct. 9 people correct. I need some more. I won't say who it is. I've got 1, 2, 3, 4, 5, 6, 7, 8, 9. We're at 9 correct. We're at 9 correct. See if you can beat the last class. See if we can get 10. Come on, there's some holdouts here. Don't be a big whiny baby and not put your guess in there. Let's go, let's go. We got time for this mediocre crap. You guys are juniors. Boom! We got 10. All right. Let's see. So let's walk our way through this. So first one, by what process are damaged cells in a wound replaced? Okay, so remember mitosis is going to be somatic cells. Somatic cells are going to be centered on growth, repair and growth. We kind of talked about that if you're a multicellular organism. So that would definitely be mitosis. So mitosis for sure, unless you have damaged cells that are replaced by sperm and egg. If I got part of my eye cut off the other day, if I had sperm replace my eyelid, that would be a mess, wouldn't it? Literally and figuratively would be a mess. Second one, by what process are eggs formed? Eggs are gametes, so yeah, meiosis for sure. Third, by what process does a zygote develop into a multicellular organism? This is the one that most people were missing. I think you were thrown off by zygote, thinking that maybe that's a gamete. That would be like a fertilized egg. And you're developing. What word could we put in for develop that would make it probably a little easier for you to get mitosis? A lot of you guys were saying meiosis. Let's go to the chat. What could you put in there instead? Abigail is correct, ma'am. And thank you for putting that direct message. Everybody should look in the chat box. It would be centered on growth. So that would be 100% correct. You would get two gametes, an egg and a sperm, that would make a zygote, and then that would grow, that would develop into a multicellular organism, and therefore this would be mitosis. Fourth one, in what process are identical daughter cells produced? Look like everybody got that one right, mitosis. Obviously, identical would matter. If you had identical sperm and egg, everybody would look the same. All organisms would look the same. There'd be no diversity or variation in the world, and it would be boring. Which process reduces the number of chromosomes? Most of you guys got that too. A few of you guys missed this one. That's going from diploid to haploid, so we would be reducing chromosome number. Awesome, you guys did great. I'm going to go to today's lesson in Schoology. Everybody should be making their way to Schoology. Go to Schoology. The most fantastic place in the world. It's where everybody wants to go, is Schoology. Cam's here. He said that's where he wanted to go. Javi, he said, hell yeah, I'm already there. What are you talking about? Let me get over there. First thing I want to do is bring up this agenda. So there is an agenda. What do we have at the end of class today? What do we have at the end of class today? Brandon, can you tell me?
We have quiz.
You do. You have a quiz. So what we're doing today, you will be quizzed over today. So we want to make sure that we are firing on all cylinders. I have also included the reading from the textbook. There are 11 of you guys that have picked up textbooks. Congratulations. Everybody that's an in-person student has been able to get them, but not all the hybrids. So let me turn around and ask. Some of you guys are hybrids. Did you check out textbooks to all of you guys? Patty did not? Patty, did you? Yeah, so you can go by the front office today. I would go at lunchtime, so I've got to go to the front office. And this is the text. This is the text for your lesson that we're going over today. So we got 2.6 is a topic, and it's pages 105 to 110. Very, very short. It's not much. I love this lesson. I think it's a fun lesson. The introduction to new material is going to be titled Stories with Pope. What is the code? So this is a pretty fun little story. Now, this story is also going to take me about 20 minutes if everybody's kind of slacking off. It'll take 15 minutes if everybody's not. I'm going to ask lots of questions of you guys. The more participation I get, the less time this could be. This could go down to as low as 15 minutes, or it could be as high as 25. I don't know though. We're going to try to keep it right around 20. Hopefully we get a lot of participation. Then you're going to get 25 minutes to work in groups, Q&A for 10 minutes, and then quiz 10 minutes. So we're going to try to stick on time. And now is exactly the time that we need to be able to move to our story. How do I get to that story? Well, let me just share my screen with you in a moment. Before I do that, I want to show you, you cannot get to everything. When we go to breakout room, you're going to be using this link, Stories with Pope. This is a link for you guys that you're going to be using when you're in your breakout rooms. I am not going to release the quiz. I'm not going to release the PowerPoint for you to look ahead so that I can get you guys to participate a lot today. So Jalen, What is the last thing that we're going to do in class today?
Quiz.
Quiz. Awesome. All right. How do I get through this in potentially up to 15 minutes? Get through the introduction to a new material. Let's go to Aspen. No, I didn't call you Brandon. Aspen, how would I cut this from 20 minutes to 15? Earl, how could I cut this from 20 minutes to 15 minutes on the introduction to new material?
If everyone participates correctly.
So the opposite of what Aspen was just showing us?
I guess so.
Yes, yes. Thank you, Aspen. Yes, now he's participating, but he's in the chat. So please come on mic so that it's easier for everybody and not direct message me. It'll make your and my life a lot easier. Okay, so let's get into the story. Stop sharing my screen. Oop. I'll be right back. Go pick up the Nizotes. This is actually a really cool story. And the coolest thing about this story is that you know more about the story than you realize. You know a lot more about the story than you realize. In fact, you know more about this story than the people who created this story in the 1920s to 50s. It's actually remarkable how much more you know than scientists at those time frames. What's cooler though, is that with what limited knowledge they had and what limited, I guess in this case, techniques that they had, they figured out the answers for this story. And this is actually a really remarkable story. So today's real lesson again is really what is the code? So obviously most of you guys are totally understanding that DNA is going to be the code for this. And code for how did I get my traits? If you ask anybody on the street, like on a late night television, and they said, hey, what gives you your traits? They're going to say, oh, it's DNA. Now, could they tell you much more than that? Probably not. Probably not. All right, a couple key points, 3 key points. DNA is a substance of inheritance, which we just had an exam about, right? We're going to inherit DNA from our parents. And when we inherit DNA from our parents, it's going to give us the traits that we have. We know that this heritable information is encoded in DNA. And we also know that there are many different, that it's going to direct not just our traits, but sometimes even other qualities outside of directly what you would consider to be a typical physical trait. It could even dictate behaviors that you have. So why is it that, at a certain age, you start to have thoughts that move away from, playing with tinker toys to maybe being more interested in sex? Well, there's a genetic component to that as well. There'll be expression of hormones. Those hormones that you can't physically see necessarily with your naked eye are going to change behaviors. And so those behaviors have a genetic basis to them as well. But this is something that people didn't generally know. Before we start this story, I'm going to stop and I'm going to ask you, is there anybody here that has a deeply held belief that they wouldn't let go no matter what evidence was given to them? Is there anybody here, and don't answer yet, think about it for about 10 to 15 seconds. Is there anything that I believe that I would not let go no matter what somebody showed me? Think about it for a moment. I'm gonna turn around and ask these kids in here. Is there anything, no matter what I did, that you would still say, hey, I still accept this even though I've showed you evidence contrary? Okay, in here, I've got 3 no's. They wouldn't, and one person says yes. Drop it in the chat. How many of you guys would, no matter what evidence is given to you, would still hold on to a deeply held belief? I won't say your name. Okay, so there are 2. There's some, I want to go ahead and tell you that's kind of a dangerous idea. You want to be open to data, be open to evidence, because when you don't question some of the things that you may or may not hold dear, you get situations where you have these conspiracy theories that people are willing to hold on to no matter what evidence is supported. It's a slightly different caveat to it, but we want to remain kind of open. But I want to also know why and what people were thinking at the time. So let's get into this. So scientists needed models. In order to test these things, it's very difficult to test heredity or DNA or trying to figure out what's going on with multicellular organisms, because multicellular organisms often have nervous systems. So scientists really had to think, what can I use? And bacteria and viruses, while bacteria are alive and viruses are not technically alive, neither of them are going to have a nervous system, and they're reasonably easy to work on, but they did employ some multicellular organisms as well, and you'll see it's going to be kind of sad. So the first one was Griffith, and Griffith had an experiment that he used pneumonia on mammals. Now, while we can't use, do labs, or we use mammals and test different things on them that could hurt them like drugs or disease, he actually could. And what he decided to use was 2 strains of a bacteria. One was pathogenic, meaning that it caused disease, and one was non-pathogenic, meaning that it didn't cause disease. So one caused a disease and one did not. This is what his experiment was like. So what he found was he did this 4 different times, 4 different ways. So he took one pneumonia that was called an S, like an S type, an S meaning here you can see these bacteria have this little capsule around them. And this capsule protects them from your immune system. They're very virulent. These are the pathogenic type. When he injected these bacteria into these mice, it killed them. No real surprise there. It's kind of what he expected. The R type, the ones that did not have any kind of, they weren't pathogenic, they did not kill the mice. So the R strains did not kill those mice. Now, he treated these S phase with heat. The question at the time was DNA the code or proteins the code? Why would heat rule out proteins? Why would heat rule out proteins? You at home, drop it in the chat. Privately, you in here, you will talk to me. I'm going to mute it. So why would heat eliminate protein? I'm going to mute myself for All right, let me go to the chat. Now drop them in the chat privately. Let's see if we can get there. Let's see if we can get there. Why would heat potentially, excellent, good job. Holy crap. Many of you guys are not only just saying how it works, but you're using the term itself, because heat breaks down, because it denatures them. Proteins get denatured, heat breaks down. Yes, okay, so that's what he did. He said, okay, I'll apply heat to these. If it's proteins that are communicating heredity, well, then the mouse is going to live. And he saw that to be true. Then this is the one that really tripped him out. He's the guy who discovered something new. When he took S strain pneumonia, the ones that had been treated with heat, and so the proteins were broken down. And remember, you can't go backwards. Denaturation cannot go backwards. When he mixed them with the non-pathogenic R strain, it killed the mouse. So he's thinking, what in the heck? Now, the thing that they didn't know, or they did know, was that DNA would reassemble itself. You can break it, but it'll go right back together after you've cooled it down. So he called this process transformation. Let me show you in a slightly different way. I'm going to show you a video of this. See if I can get to the video, hopefully. Abracadabra. Let's get to it. I wanna be Johnny Brown. Where's Earl crying? Earl, yeah, you're supposed to be taking.
When you
this-
find the perfect engagement ring at Hellsberg Diamonds, you feel like a champion. And Patrick Mahomes gives you a personal shout out.
After he loses the Super Bowl.
Hi, bad people. Today we're going to discuss Griffiths and also Hershey and Chase. Both of these experiments were very important in discovering exactly how genetic material can be passed on from parent to offspring. Griffith's experiment happened first in 1928, and so we're going to discuss that one first. Griffith's experiment proved that bacteria are capable of passing on genetic information. Bacteria are capable of undergoing something called transformation. He thought that information might be passed between organisms which might cause them to assume a different form. Pneumonia was a major cause of death at the time of Griffith's experiment, and so he chose to experiment using the pneumonia bacterium. Griffiths also knew that there were two different strains of the pneumonia bacterium. The rough strain, or the R strain, was relatively safe, whereas the smooth strain, or the S strain, was pretty dangerous and almost always killed the subject. Normally, anybody infected with the rough strain would survive, and anyone infected with the smooth strain would die. Griffiths tried 4 different combinations. He first injected the R strain into a healthy mouse and the S strain into a healthy mouse. Predictably, the mouse involved with the R strain survived. The mouse who was injected with the S strain died. This was fairly predictable, but the next 2 steps show why Griffith's experiment was groundbreaking. Remember this structure from the fall? This structure that we drew way back when represented an enzyme, which is a type of protein. Notice that it has a very specific shape. If the goal of this enzyme is to put together these 2 substrates like this, it must have this exact shape in order for the substrates to fit nicely. If this enzyme or protein is subjected to any kind of heat, cold, acid, or force, it will change its shape. Now, even though this protein still has 2 openings, or activation sites, the substrates can no longer bind to it the way that they should. Remember also that we call this process denaturation. Griffiths hypothesized that the specific shape of a bacterium would allow it to do something very similar. Using its very specific shape, the bacterium would be able to bond to other substances inside the body. However, if you expose it to heat, cold, acid, or force, the substrates would no longer be able to bind to the bacterium, and the bacterium would not be able to function normally. Griffiths exposed his bacteria to the heat source. When exposed to the heat, the bacterium changed their shape. Of course, he wasn't able to see the bacteria change its shape, but he was able to conclude that this is what had happened. When he took the heat-killed S strain, which used to be deadly, and injected it into a mouse, amazingly enough, the mouse survived. Heating the S strain bacteria had caused them to become denatured, so they no longer were deadly. Griffiths then combined heat-killed S strain with live R strain. Remember, R strain bacteria are not considered to be deadly, and heat-killed S strain bacteria are not considered to be deadly. However, when he combined the 2 of them and injected it into the mouse died. Griffiths hypothesized that even though the S strain bacteria had been denatured by the heat, that there were still components of it that were still active. He thought that the components of the S strain bacterium were going into the R strain bacterium and corrupting it somehow. This could cause what was previously an innocuous R strain to become a toxic R strain. We call this transformation. Now let's talk.
Now, what's cool about this is that students that are actually in class will be doing an experiment to test this in about 3 weeks. We're going to actually transform bacteria. We're going to give them, we're going to be giving those bacteria a gene on a little loop of DNA and change them. Well, let's get back to it. You guys can see this PowerPoint now again, yeah? Okay, cool. So, question is, at this point, do you think people accepted DNA as a code? He showed, in this case, Griffith showed, well, proteins are not the code, right? Proteins aren't doing this. It can't be proteins because I denatured the proteins, yet they, and these mice died even after being given a mix of non-pathogenic and denatured pathogenic bacteria. Do you think people accepted this? Drop it in the chat. You can keep it open or direct. I don't care. It doesn't matter. This is all just what you think people did anyway. Not straight away. Yes, no, no. Yeah, give it just to me. Be brave. Share this with everybody. I think it's kind of important. Some of the things that you're saying. No, they understood the experiment, Tracy. They totally understood. They were showing this to other scientists. Other scientists definitely understood this experiment, for sure. But a lot of you guys are saying no. Now, the thing about that is one of you even said, well, from the tone of your voice, I would say no. And that's pretty good. You're probably got really high emotional intelligence. You're right. Culture changes slower than anything else. Nothing is slower to change than culture itself. Within a culture, the culture does not change. Even scientists who pride themselves as being the most objective people struggle with this. This is why we had the scientific method, to try to eliminate any kind of bias that we have. So no, they did not at first. There was a lot of people who had staked their careers on proteins. If somebody told you something you'd been studying for 30 years is a bunch of crap, then you'd be like, I don't know how I feel about that. And so logically, it makes sense, though. I want you to think about this for a moment. I want you to reject this idea. There was still some logic behind this. Think for just a moment. Proteins or DNA, which is the code? How many nucleotides are in DNA? Drop it in the chat or come online. How many nucleotides are in DNA? And you can text this out to everyone. You don't have to keep it just to me. You don't have to, yeah, okay, there you go. You're keeping it just to me. Drop it out to everybody. There you go. Thank you. You're right. 4. Yes. There are 4. Very straightforward, not too difficult. Guanine, cytosine, thymine, adenine. These are our 4. But how many amino acids are in proteins? I haven't taught this to you. How many do you think it is? How many protein or amino acids are in proteins? Just take a stab. We know they're extremely complex molecules. Just throw a hazard a guess. Somebody's being a smart butt and looked it up. Thank you. Yes, thank you guys for taking a chance, though. Mock, I admire you for throwing that out there. Yeah, it's 20. It's 20. Now, what is going to give you the most opportunity to make things complex? 4 or 20? So you can see these are all, and you don't have to memorize this. These are all the different amino acids. So I can either take 4 things and come out with an organism, or I can take 20. It's pretty straightforward. How many letters are in the English alphabet? Let me ask somebody in here. Javi, how many letters in the alphabet? 26. You are correct, sir. There are 26. Think about this for a moment. There's your biology book, Biologia. Here's the Bible. Here's Harry Potter. From 26 letters, I can recombine these letters into any word and rearrange them in any order to come up with any sentence to give you a nonfiction text, a theological text, I guess that's what I should say, and then completely fiction. I can tell any story I want with 26 letters. 26 is greater than 20 and a far might greater than 4. So it kind of makes sense that scientists want to stick with 20. After all, 20 versus 4. What makes more sense? An organism, look how complex and cool we are. There's our muscular. That is sort of a person. We could consider him to be also AI. What y'all's guess? Is he AI or is this a human? AI, oh yeah, AI. Okay, other people are saying human. Other people are saying AI. Abby, why do you want to say he's AI? Tracy, why AI? What do you guys think? Oh, he looks scary. Aspen, Elon, a whole robot. Thank you, Aspen. Yeah, Elon Musk, who knows, right? This guy's, he may be superhuman. He may be integrated with a machine. He's robotic for sure. But get back to reality for a moment. We got a lionfish, pretty elaborate creature. Insects, as small as they are, fairly elaborate creatures. But plants, variable. Look how variable these plants are. Or it could be as simple as a bacteria. Simple to complex. What makes sense? To have 20 different pieces of information to make all this complexity, or 4? What would make more sense? So it was logical for people to think this for a moment, to reject this idea for a moment. But is it so logical? Now, I want you to consider how many different symbols does it take to make this? Don't answer in the chat yet. The image on this television, the image on this phone, TikTok. Don't answer yet, guys. Come on, hold on. YouTube, the image on this computer. How many symbols does it take? Now you can drop it on the chat. Let's see what we get. Chat to everybody. Don't be scared. Okay, good, good. Several of you guys are saying it. Only so far do I only have 4 correct answers. You're right. Some of you guys are right. You know that it's only a simple number 2. Only 2. How complex is a video, is a movie, is all the information on the computer? How complex is that? It's extremely complex. What you're looking at right now is comprised of only these 2 symbols, a 1 and a 0. It's binary code. Now, there are only how many types of people? Only how many types of people? Not 10. 1, exactly. There's only 1 0. There's only 2 types of people in the world that understand those binary code. Those who do and those who don't. I'm one of the people that do not. I am not a computer programmer. I have no idea. The point that I'm getting to you here is this. It's not a stretch. If I can, with only these 2 symbols, I can get this. Why not? With only 4 symbols, can I get this? I mean, can I not get this? I can get this with 4 symbols. After all, that's 100% more information than just 2. So it is possible. Okay, last thing, and then we're going to go to our breakout rooms. It looks like I have 11 seconds. So we're going to go to the last experiment. I'm going to stop sharing my screen. I'm going to go back and finish our video. Hold on. This alarm's going to go off. There we go. All right, let me go back and to my video. And let's see, what did we do next? This is a classic.
Talk about Hershey and Chase. At the time, most people thought the molecule of heredity was contained within proteins. Almost no one believed that the molecule of heredity was nucleic acids. Proteins are so complex that it wasn't that big of a stretch to imagine that there might be a nose-shaped protein, or a height protein, or a personality set of proteins. There are approximately 150,000 different proteins in the human body. Remember that proteins are made up of 20 different amino acids, which are arranged in very complicated patterns and shapes. This leads to a huge number of potential combinations. Nucleic acids are much simpler. There are only 4 different kinds of nucleotides, and there are only a few different types of nucleic acids, DNA, and a few different types of RNA. Hershey and Chase set out to prove which of these 2 things, proteins or nucleic acids, was responsible for passing along genetic material.
Grammarly can help you write quickly and confidently, so you never have to slow down at work.
They must know I'm a teacher. I need Grammarly.
Instant corrections make your writing mistake.
They chose to do their work using bacteriophages. Bacteriophages have several different advantages, including the fact that they reproduce very quickly and are only made up of 2 of the big 4, nucleic acids and proteins. They don't contain any lipids or carbohydrates. So here we can see that this bacteriophage has a protein coat on the outside and then nucleic acids on the inside. Bacteriophages reproduce by attaching to the outside of a host cell. Then they inject their genetic material into it. The protein coat stays on the outside of the host cell. The DNA from the bacteriophage then causes the cell to stop whatever it was doing and begin producing copies of the bacteriophage. The new bacteriophages are identical to the parent bacteriophage that originally infected the cell. The host cell then explodes, releasing all of the new bacteriophages, which then go out and infect even more cells. Hershey and Chase used this life cycle to prove which of the two contenders was actually responsible for inheritance, nucleic acids or proteins. Hershey and Chase used two groups of bacteriophages. In one group, they chose to test the protein coat, and in the other group, they chose to test the nucleic acids. To test the two options, they decided to make parts of each type of bacteriophage radioactive. They knew that whichever part was responsible for genetic inheritance would create radioactive baby bacteriophages. For the protein coat, they made the protein coat radioactive. Since proteins contain the element sulfur, they decided to use a radioactive type of sulfur. The type of sulfur they used is called sulfur-35. Since sulfur is not found in nucleic acids, they were only marking the protein coat. For the nucleic acid group, they decided to replace the naturally occurring phosphorus in the nucleic acids with a radioactive type of phosphorus called phosphorus-32. Since there was no phosphorus found in the protein coat, the only thing they were marking in the second group was the nucleic acids. Hershey and Chase then allowed each type of bacteriophage to infect a host cell. They knew that whichever type was responsible for inheritance would create radioactive bacteriophages. The bacteriophages then injected their genetic material into the two host cells. Remember, the protein coat is left on the outside of the host cell and only the DNA gets injected. Hershey and Chase then shook the compounds in a blender to shake the protein coats off the outside of the cells. The protein coats then dissolved in the liquid portion of the experiment, which is called the supernatant. So now each blender has a liquid portion which contains the dissolved protein coats and a solid portion that contains everything else. However, at the moment they are mixed together in the blender, and so we need to separate them. To separate the liquids from the solids, the scientists then put the mixture into a centrifuge, which spins very fast and separates components based on density. After spinning the components in the centrifuge, we now had a liquid portion containing the protein coats and a solid portion containing everything else. We call the solid portion the pellet. Hershey and Chase then tested both the pellet and the supernatant in each case to determine which of them was radioactive. Remember, the goal was to make radioactive bacteriophages, which would be found in the pellet. In the situation where they had tagged the protein coat, the supernatant was radioactive. However, the pellet containing the new bacteriophages was not radioactive, and so they decided that that must not be the molecule of inheritance.
Yeah, what I think is fantastic about this experiment is just how simple it is. It truly is remarkable that they were able to answer this question with something that with such a simple technique but you had to be very creative to actually think about how they were going to do this. Guess what killed Martha Chase? This is one of the famous biologists. She really did a lot for women in biology. Just what do you think killed her? There's a little bit of irony here. Science did not kill her. Science never killed anybody. What do you think killed her? Not bacteria. It's a good guess. It was bacteria. It was, but what specific bacteria you think killed her? Not radioactivity. That would have been even more. There'd been more irony. What do you? Earlier they did. What do you think killed her? My students and I, there you go, pneumonia. Several of you guys have just said pneumonia. I was teaching at the time that she died, and we actually took a day just to spend discussing her prior to ever even getting to this topic. She was a magnificent woman. She was really a magnificent lady. I remember learning this as a child thinking, oh my God, what a brilliant woman, and just being so impressed with her. Okay, everybody's going back to Schoology. Everybody's going back to Schoology. It's where everybody wants to go. You're like, oh my God, I woke up this morning, where am I going? I've got to go to Schoology. Oh my God, I'm so excited to be there. Please go to Schoology pretty quickly, and we'll meet you there. Let me just go ahead and just jump from the video. We'll go to our heredity exam. That's where we want to go. We don't want to go there. Let's hold on. Let's go to our class and courses. Now everybody has the same document. I'm waiting for everybody to get to, oh, I'm keep going to the exam. Sorry, keep going to the exam. We're going to what is the code. Here's the document. I'm going to go ahead and open up this PowerPoint in case you would like to reference it. The PowerPoint is reference. Everybody needs to open up Stories with Pope. What is the code? Let's see who's already there. Daisy, we're waiting on you. Jessica, Precious. Precious is already there. She's as fast as a lightning bolt. Woo, Precious. Okay, so when you get there, you're going to see that there are both of these experiments are there. I have gone ahead and given you kind of summarize this, and then I've asked 5 basic questions. You're not going to just be able to look this up and think about it. You're going to have to actually think about what we've discussed and what I've written for you here. And then you're going to answer these 5 questions. Now, I was going to give you 25 minutes. I do not have 25 minutes to give you now. I'm going to only be able to give you guys 15. Sorry, that's probably going to be enough. So I'm going to open up breakout rooms. You'll go there. You're going to do the best you can with these questions, and then we'll come back and discuss them. So get ready. You guys are gonna stay in here and discuss it. You guys don't have to go to the breakout rooms. You guys have each other. Y'all are your own breakout room. Okay. Get ready to go. The air open. Mr. Brinkman, do you have any questions for me before I start working with the students in here?
No, sir.
Okay, I'm gonna go and work with the students in here. Okay, all right, guys. Let's see. First things first, you really don't need this. This is right here. That's what you're gonna be doing. So you need to go to a breakout room. You guys discuss this openly here. I'll mute it so people can't hear through there. And go through this. And if you have any questions, I'll be able to assist you along the way. They're going to be on their own. Mr. Brinkman, did you happen to go visit any of the rooms?
I did not. I found that when I hop into rooms, I tend to be a disruption to the learning in the small breakout room. So
Okay.
I've
All right.
taken that off of my menu.
Okay. I don't think you'd have squelched these guys too much. Honestly, I don't think so. So if you ever come back, it'd be interesting to have a fly on the wall. I think I would probably be more obtrusive, but they'd probably be like, somebody else.
Might be.
We'll see. We'll have them all back in 4 seconds. We'll have them all back in 4 seconds. Welcome back, Carter. Let's see. Would anybody volunteer us to use their document? And then we'll compare it to mine. Would anybody like to volunteer to use their document as a walkthrough, or would you guys, are you guys too timid? I'm sorry?
I don't mind.
Okay, could you tell me who, is that Tracy?
No, it's Mia.
Who is it?
Mia.
Mia. Well, Tracy's always the most verbose kid. That kid talk to you about anything and everything. All right, let's go see. Wallace. Mia Wallace. Queen of the Scots. All right, let's see. So let's see what you got here, kiddo. Oh, I had 4 more volunteers. Sorry, thank you. I didn't see you. So let's see what you guys. What was the independent? You said the independent variable was the strain. I agree. The dependent variable was the mouth. Now, that being said, I do agree with you, but there was more than one experiment conducted. What would you say was the other independent variable? Because, okay, let me back up for a moment. Mia, you say independent variable was strain and dependent variable was the effect it had on the mouse. What do you mean by effect?
Basically, whether or not it died.
Okay, whether or not it died. Okay, what was the question? Because we always start with the question, then we come up with a hypothesis. So essentially, you just gave me this hypothesis. If this strain, then death. What do you think the question was?
Wait, hold on, let me go back up to it and see if I can derive from what I am given.
Well, what was the debate that was being had?
DNA. It was about DNA. The structure of
DNA or what?
and the function, instruction.
Nope. That's next class. DNA or what? What held the code? DNA or what?
RNA.
Nope.
Oh my God. Amino acids.
Amino acids make what?
Proteins? Oh, proteins.
Proteins.
Yeah.
Okay, there we go. So is it DNA or proteins? Cool. So the follow-up experiment, he had a new independent variable. What was that? And y'all can help this poor child. Jeez.
Please. Oh my God.
I know. They're like just leaving you hanging out there. Remember that when somebody tries to get you to go to prom with them next year, you'd be like, you left me hanging. What was the subsequent variable? We had here, R strain versus smooth strain. What was the next experiment?
Heat.
Okay, heat to test what, Jalen?
To test if the heat if using heat would kill the smooth strand and allow the mice to live.
No, we knew heat would kill the bacteria. The question was whether heat was it was going to be.
Wasn't it whether or not the heat will like mess up the proteins? It was trying to see that if the proteins no longer if the protein is messed up and it can no longer do stuff.
Let's not use messed up. Let's use a little bit academic vocabulary. Denatured. There we go. Now we got the fog. Now the fog has been lifted. There we go. So there was really another experiment, right? So which really answers our second question, right? I mean, we already got the second question. So this was a pretty cool experiment that started to sway people to thinking, oh my God, DNA actually might be it, rather than proteins. And the most people at this particular moment in time, this was literally 98 years ago, thought proteins were the answer, right? Proteins are obviously it. So let's go to see what you get for Hershey Chase, kiddo. So you said, based on the Hershey Chase results, describe how this bacteria, okay, so you said by blending and replicating itself sneakily. Nope, I'm not going for that. Somebody help this child. And thank you for being so oh, hold on, there was a subsequent question 3. Oh yeah, why? Yeah, what was the word we're looking for? There's a word for number 3. I'm really looking for a word for number 3. It starts with a T.
Transformation.
Transformation. And add this to your vocabulary. You're going to need that. We're going to do, this will be our next IA, is going to be about transfer. The kids in this classroom are going to take DNA from a jellyfish and insert it into bacteria in 3 weeks. You guys are going to do that in this lab right here. So it's in that refrigerator right there. It's going to be tough. You guys are going to have to put your big boys on. Okay, so number 4, help her with this one. We only have 6 minutes. No. Yes, we have 6 minutes, and I've got to release you to the quiz. Because I don't think you're really addressing the question here, sweetheart. I'm looking at this. It says, based on Hershey Chase experience, describe how the virus was infecting the bacteria.
I don't know.
Well, look up here and see. How was it doing this? Look at the image. Take the diagram. What was it doing? Oh my God, would y'all quit texting me privately? Text it to Mia. Can you send this information you guys are sending to me to Mia as well? Or are you not allowed to do this? Can you guys message each other in here? I don't know. Can you try to message somebody and see if you can do it?
You can.
Okay. Then why are you sending this to me? I know the answer. Help this child.
It injects its genetic material into the cell.
Yeah, you're right. It's injecting. What is this genetic material? That was the question. Was it protein or DNA?
DNA.
Okay, Jesus, we're finally there. My goodness. Now you guys got stuck on this one, so I'm going to share mine. This is Mr. Pope's answers. I did this while you were doing this.
Mr. Pope, we can't see your document.
Oh my gosh. Could you see Mia's? Oh, y'all couldn't see Mia's either?
No, we could only see Alosa's.
Well, why didn't you tell me? Oh my goodness, you should always say it. Now can you guys see mine? Well, that was a big fail on my part. Okay, here's mine. So how did I write this? Now, this particular one I really want to be careful about, because just like on y'all's exam where people are losing lots of possible marks, if it says with reason, right? So based on the results, did DNA or proteins carry genetic information? If you say DNA without giving me a reason or giving IB a reason, it doesn't matter. So in this case, you must include qualitative data. So there's no numbers up here. Up here though, you can see, and in the experiment, you could see that DNA that was radioactive with sulfur, the sulfur, the phosphorus was actually inside the bacteria. The sulfur was outside of the bacteria. So the bacteria that was radioactive with sulfur, it transformed. And if it, and it was therefore DNA had to be the answer to this question. This is what convinced the world that DNA actually was transferred heredity. This is what you would need, and very close to how you would need to say it in order to score that mark, in order to get that point. I'm not using one sentence. I'm given a reason, and therefore I'm going to score that mark. If you just say, well, this is the answer, nobody cares. What they do care is your ability to not only do that, but to also give the reason for it. Okay, I'm going to open the quiz. And I'm assuming you guys can see you guys, next time you've got to help me out. If somebody says, hey, man, I can't see that. I'm going to open the quiz. Everybody should be able to see the quiz. That is your exit ticket. And I will see you Wednesday. Wednesday. By the way, did your advisory teacher tell you guys about asynchronous classes on Friday?
Yeah.
Oh my goodness, don't yeah me. Okay,
What?
you guys have 5 minutes. If you'd like to talk to me about the asynchronous classes, go to your advisory teachers or come to me at office hours at 3:15. In the meantime, deuces. Once you take the quiz, I'll take attendance from the quiz. I will see you on Wednesday, if not at office hours today. Quiz is right there. What is the code quiz?
We can leave.
After you take the quiz, you may. By the way, 86, that was the average for first period. If you guys can beat 86, we'll give you an extra, I'll give you an extra 1 point if you can beat first period. 86 was the average. They killed it. Nobody knows. Let the fire get higher. Everybody so far, 100. Is Terry not here today? She was actually physically at the school? Shit. I'll have to post this lesson. If you're done, you can split. Make sure you play in the street tonight, though. Congrats, most of you guys made 100 on this. Wasn't hard, this is an easy one, that's all right. Woo. The abracadabra. Get up, your mama can't cool down. She's getting running round and round, yeah. Round and round and round and round. You can't play in the street. Don't get timed out on because you only have some of you guys 1 more minute. I can see your time's running out, yo. Some of you have 2. All right, I'm gonna close the call. You guys be good.
Bye, Mr. Pope.
All right, bye-bye, Plate. You guys have a nice day.